Thank you, Michael. I will be talking about the bioprinting of blood vessels today. Uh, we'll start with a bit of a background discussion and background about why we are doing this work, starting from the basic preclinical research to clinical research, specifically looking at what is a traditional drug discovery path look like. So if you look at the drug discovery path itself, it spans over a period of more than a decade. Where 2 to 5 years you'll be spending in a basic science research, followed by 1 to 2 years in preclinical research, looking at animal testing. And once we refine that, you will enter into human trial, which will take you anywhere between 5 to 7 years. Followed by then you have regulatory approvals and governmental approval processes, which can last up to 2 years. Overall, adding up to more than a decade and a half before your product goes to the market. So it's like a funnel, as you can see here. If you start at basic science research with about 10,000 compounds, you will end up with 250 by the time you research to an animal testing stage. From animals testing, by the time you want to enter into human trial, there will be only 5 compounds. And towards the end, through the whole process, 1 compound will be left. So starting with 10,000, you're going with 1 compound in up to 15 to 17 years. Estimated $2 to $3 billion, which is a large amount of money invested on a failed drug. And you can see why. 9 out of 10 clinical trials also fail. So there is a need that we need to refine some of these processes. They're definitely not ideal because we're spending a lot of time and with least amount of efficiency. So let's look at what the basic stage and the preclinical stage look like and where we're going wrong. So when we look at the discovery stage, which is lasting anywhere between 3 to 5 years, predominant work we do in terms of drug discovery happens on two-dimensional cell culture. They lack multicellular interactions. They don't have the complexity of the 3D tissue, which is present in vivo condition. When you look at the preclinical stage, which is mainly looking into animal testing, this is what our current profile look like. So from the discovery stage, you are testing your inventions into small animals, going back until large animal before it enters to humans. And more than 115 million animals are used per year in research. These animals do not replicate genetically as well as physiologically the human diseases. So they are unreliable, they are very expensive, they are very time consuming and resource intensive. This is just a graph showing you a type of animals which are predominantly being used. If you look at mice, is the most predominant. The other mammals, they're actually less, but as the size of the animal grow into preclinical testing, your cost also increases. So there is a need to refine the basic and the preclinical research in order to be more proactive and shorten this path, which has an implication on the translation, at the same time, the cost of these developments. For that purpose, we need a fast win and a quick fail approach. Bioprinting is one such technology, which can offer us lots of possibilities in order to get something in a 3D, which mimics the in vivo conditions of the tissues. So for those who are new to this technology, briefly I will introduce you. Bioprinting you can define as a synchronous positioning of biomaterials with living cells in a prescribed layer by layer fashion in order to organize or fabricate a live tissue construct. With cells, you can mix them with proteins, with DNA of your choice, drugs, growth factors, other bioactive substances in order to guide tissue regeneration or formation. So there are various types of bioprinting technologies. Predominant or most commonly used is extrusion-based bioprinting, which is in the center. Discussion about what bioprinting different technologies out of the scope of today's talk. But if you are interested, you can refer to my book, which is on 3D printing and medicine. So what does the opportunities these 3D bioprinting technology offers to you? It's a huge opportunity for personalized medicine. The reason is if you are able to extract patient's own cell, you can create personalized tissue and thus develop bespoke therapies, starting from developing bespoke or precision personalized model to test different types of drugs, which are efficient enough for patient-specific application. This also has an implication of reducing animal testing. We can also understand by taking biopsies from patient to develop patient-specific disease model in order to understand their diseases. And if we are successful in all these two stages, we are then going towards our ultimate objective of creating tissues and organs for transplantation. So overall, bioprinted tissues in a preclinical research as an opportunity, instead of going from the current route of entering into human trial, we may be able to do this, where will be our future approach. From discovery, we will be able to utilize these bioprinted humanized tissue models to refine our and test our technology for a certain efficacy before we get into human trial. And the reason this is important because it actually provides you human relevant tissues for testing. It does have a complexity of an in vivo environment, helps you to get targeted screening, reduce the time frame for preclinical research, and ultimately reduces the animal usage we are actually doing in preclinical research, heavily dependent on it. And overall, the research becomes very ethical. So my team as such works in this area for more than 2 decades now. And we also run a platform called 3D Bioprinting Research Platform. It's an international platform funded by British Council. And we have been collaborating with a lot of different organizations during this process. There are 3 components which we are researching. The first one is innovative bioinks, where we develop different types of bioinks for bioprinting cells, which we also call it the holy grail in terms of success of bioprinting. Then you have a hardware, which is bioprinter. We work with various industries to develop these various different types of tissue-specific hardware for bioprinting applications. Along with that, we are also exploring various medical imaging technologies, which are essential part of this workflow, whereby you can integrate them with the bioprinting technology, so that in future we are able to generate the organs and the tissues where from the medical imaging itself. This development of novel bioink, which, as I said before, is the holy grail in terms of success of this technology, where you mix cells with these materials so as to they can be printed. We specifically focused on synthetic biomaterials because of their consistency and reproducibility. They're free from animal origin. They're easy to process for bioprinting application, and we can tailor their physical, chemical, as well as biological properties. These are some examples where we have printed various different types of tissues, tissue architecture. They have different types of properties apart from just printing. They have a different rheological properties. They can be stretched. They have a self-healing properties. We can tailor them. That allows us to print them into various different shapes and sizes. Biometrics is one such product or a bioink which we have produced. It's currently has a patent on it. And vascularised is another hydrogen material or a bioink material which we develop in our lab, which I will be talking to you about the current application of bioengineering blood vessel. So what are we using these bioinks for? Predominantly, my lab has been investigating development of tissues, both for disease modeling as well as for transplantation, for translational application. And we are looking into both hard as well as soft tissue. So we are looking into bone, blood vessels, skin. We collaborate with external partners on artificial cornea development. We have done a lot of work on cartilage, and we very recently completed a work on developing osteochondral model using synthetic bioinks, which we recently published in International Journal of Bioprinting. But for today's topic, we'll focus on bioengineering blood vessels. Now, why do we need to engineer a blood vessel? So if you look at the cardiovascular disease from a WHO point of view itself is a huge disease burden worldwide. It's not just restricted to United Kingdom. We all are suffering with it. And specifically looking at if when We don't even understand quite a lot of these pathologies, how the cardiovascular diseases are actually developed. There are lots of different reasoning. There are no specific drugs which actually effectively works in majority of conditions. So that itself is a new line of research which require investigation, but there are lack of models to test those drugs on. But again, that will be a part of separate talk where we can look into disease modeling specifically for cardiovascular diseases. Today, I wanted to talk to you a bit more on translational part. Is when the drugs and the medicines do not work in cardiovascular diseases, specifically looking into coronary artery related disease, we have to go for surgery. And within the surgical toolbox, there are two gold standards. One is autograft, where you take a blood vessel from same patient, but from a different part of the body, if it is available to spare. If we can't get that, then we will have to go for a synthetic graft, which is predominantly made from PTFE, polyethylene terephthalate. And it's a synthetic, which is prone to getting plaque formation and clogging. So that means it gives you some solution, but it's a temporary solution. The condition can reoccur, the blockage can reoccur. So an ideal solution would be if you have a bioengineered blood vessel. So this is not a new concept to think about whether a bioengineering blood vessel to be developed. We were not the first group to think about it. However, we don't see those vessels anywhere in the market. There is not has been much of a discussion on that one or a progress in terms of translation. And if you look at the challenges itself, there are two main challenges. First, the technology to easily fabricate cellularized blood vessel construct, which includes both the materials and the technology to get these vascular structures ready with right mechanical property, biological properties, and at the same time, the right composition, layering of the blood vessel cells within them in an easy form. The other one is the time to fabricate and mature these vessels. So just to give an example, is that relates to the access to the raw material, which in this case is cells. We need plenty of cells. So the blood vessel itself is composed of, or I would call this, is a composite tissue. It's not a simple organ. It has actually got endothelial cells, muscle cells, and then you have fibroblastic or connective tissue layer on top. So we are talking about more than two different types of cell types, which require to be engineered and placed in a very hierarchical fashion, which exists in a live condition. But to access this type of cells, primarily from an autologous source, is very time consuming. The people who work with endothelial or smooth muscle cells, they know if you have to expand them in vitro, you can spend up to 4 weeks for them to grow into enough quantity so they can be utilized for fabrication. And based on the fabrication technology, it could actually take you 1 to 2 days or more to assemble them into a layer whereby they have architecture similar to blood vessel. And it can take you anywhere from starting getting cells till manufacturing and maturation. It might take you up to more than 2 months to get your blood vessel ready. If you talk to your clinician and you explain that to them, they will say, I'm sorry, this might be an interesting product you produce, but it will be not a viable solution for your patient because patient can't wait for 2 months. They're looking for a few days and weeks by which you want to provide this kind of intervention. So this is where our research question started. How do we address these basic questions and basic challenges? And this is what we actually thought about. So going back to what I explained, blood vessels is actually a composite tissue. That means you got the inner layer, which is also called tunica intima, coating of an endothelial cells, followed by smooth muscle cells. And then you have connective tissues or a fibroblastic layer on top. Now, to assemble these into a hierarchical fashion, it itself is a challenge. We can, as I said and explained, if you try to access primary cells, it's quite difficult. It takes more than 4 weeks to actually get enough cells for any fabrication technologies to create the working blood vessel in a shortest period of time. So primary cells definitely is not a source which we can rely on. They grow very slow. The other option we got is to use stem cells. Within stem cell, we have two options. The bone marrow derived stem cells, which are routinely used in a practice in the regenerative medicine, but they're very invasive in terms of extracting them from an autologous source from the same patient. And not many patients will be happy for an invasive procedure. So obviously that restricts us from not only from technical, but also from a patient point of view. However, adipose-derived stem cell or the fat stem cells, they are much easy to isolate. I'm based in plastic surgery unit, and we get them all the time. And you speak with your patient, everyone is happy to have their fat donated, right? So in that case, we don't get it. It's relatively very simple and easy process. So that's the reason we actually hung on to see whether there is a potential to use adipose-derived stem cells to produce our blood vessels as a source. And also there is a lot of literature which suggests that fat itself has all the factors which are necessary to encourage wound healing and eugenesis. So there is a background in there in the literature which says that this might be a viable source in terms of producing our blood vessels which we require. So what we did in this process, this is a workflow of what we were looking at. There are two parts of this schematic. The first part is to get the adipose-derived stem cell. You isolate them, but you differentiate them into endothelial cells. So you create a bank of endothelial cells, a huge amount of them, up to a 10 million, starting from a million within a week by using a pod delivery growth factor system, which is what we use in this case, which delivered sustainably VGF. Whereas the second method actually looks into differentiating ADSs using TGF beta into smooth muscle cells. And eventually we combine this together into a vascular structure, which essentially is perfusable and can be available for implantations. So that's generally what our concept look like. This is again in this process, carrier of these cells and holding them in a shape is very important. For that matter, we selected our material, which we developed in our lab called vascular biometrics. The reason we choose because this is biocompatible. It has a very high shape fidelity, and it has tailored mechanical properties. So what we do, we use a coaxial printing technology in this case. So what happens in coaxial, you have two coaxial needle. The outer needle contains your material along with the cells, predominantly smooth muscle cells, giving you the outer periphery and the thickness of your blood vessel wall. And once that's done, the inside we have a degradable material. That degradable material eventually goes away, and then you have a hollow conduit or a cylindrical structure along with smooth muscle cells. Over a period, what you do then after same day, actually, you can inject the differentiated or produced endothelial cells from these ADSCs, and we inject them inside. We give them some retention time, and eventually they coat the inside layer of the vessel itself. I'll show you some picture how they look like. So essentially, this is a laboratory setup. This is a printer where we have a coaxial needle actually being set up. And then hopefully you'll be able to see that we also have this needle, they're perfusable. So you can actually perfuse their hollow. This is a scanning electron micrograph we're looking at. And looking into the size and the thickness, the wall thickness. This wall thickness is about 250 micron in this case. And you can make them as long as you want. We have went up to 15 centimeter at a time. They are quite reproducible by using this technology, where you can have a fixed inner diameter and outer diameter. The lumen can be very much tailored based on our requirement and type of vessel we want to actually manufacture. Obviously, there is a lower limit and upper limit we can go currently, but it's quite tolerable. That predominantly depends on the type of needles we are able to access. Now, coming back to the main part, is okay, did we actually achieve the assembly of the cells as we require in a natural state, which is an inner coating of endothelial cells followed by smooth muscle cells? So to do that, we actually did a live tracking of our cells. And we label our endothelial cells in a red color, whereas the smooth muscles in green color. So these are individual images looking into the inner wall or the lumen of the vessel. And then this is the next part, apart from the lumen. So the green is smooth muscle and the red, this is a combined image, showing that the assembly process did work. So the way we have been assembling, this is a picture taken after 24 hours, and you can see that there is an endothelial cells there, and then you have a smooth muscle cells layer. We mature this over a period of time. We monitor our vessels from 7 days to 14 days to see what's happening. And if these cells in this co-cultured cylindrical tubular structure maintains their phenotype. So this is a schematic. This is how ideally you are looking for your cells to organize and maintain in a tubular structure. And when we look at it, you can see that the endothelial cells are there and the smooth muscles are there. This is a day 7 picture, and then this is a combined image. Later on, we looked into their specific markers, specifically for smooth muscle cells is alpha S7 and CD31 for endothelial cells. And we can see these cells actually maintain their markers between 7 to 14 days. And then we also looked into extracellular matrix proteins such as collagen 1 and elastin, which are very important, which provides the elasticity to the blood vessels. And we can see that the cells are actually producing and also expressing these markers as the time progresses. In addition to that, the cells actually in a combined state maintaining all their individual markers. So that means there is maintenance of individual cell phenotype while they are in a coexistent tubular structure, which was very important for it to be called a blood vessel. Later on, what we did is we implanted this into an in vivo test in a CAM assay, which is a chick embryo assay. You can only maximum do this assay after implantation for 4 days, and that's what we did. Within 4 days, when we had the whole composition, including the ECs and MSCs combined vessel, we actually saw there is a lot of microvasculature coming from the CAM on top of these vessels. Now, what this assay actually tells us? It tells us that if we implant our vessel into an in vivo model, into another animal model, it will hopefully will be compatible. It will have a in vivo compatibility. It will integrate with the host tissue. And that's the maximum information you can draw from these pictures. But apart from the biological test and the maintenance of their phenotype, I think the most important thing is their functional test, whether they're mechanically robust, whether they have the mechanical property and the hemocompatibility itself. So what we did is we looked into the mechanical properties of these vessels, where we did burst pressure, which is a very common test you will do if you are looking into manufacturing of vascular grafts. So this is a cellularized vessel. And as you cellularize, the mechanical properties are higher. And as you keep maturing that vessel over a longer period of time, up to 14 days, the mechanical properties gradually start increasing, which I think is very much now explainable from the previous picture where I showed that the collagen and the elastin is actually incrementally produced by our cells, which shows that the cell itself now taking over the matrix and providing it that mechanical strength which is required. In addition to that, these vessels also hemocompatible because this material itself as a vascular biometrics do not actually allow attachment of platelet. So we compare that with vasculometrics against PTFE, which is a standard control at the minute. And we can see there are a lot of platelet attached on this compared to vascular. So that's again a couple of test paths where we can say this is a compatible material for these applications. So in summary, what we learned is that we can definitely address some of the challenges we had in terms of accessing the primary cells by using adipose-derived stem cell and then using a clever systems such as growth factor delivery systems such as POTS to differentiate them into functional vessels. There is definitely a clinical potential for these technologies to be addressed. Most of the work we have done in this case is in the static condition. So it needed us up to 1 week to 2 weeks to get a mature vessel. Once now our future work will be focused on dynamic conditions where we will be flowing, we will be putting them in a bioreactor, and we hope to reduce this time frame even further. So that's the work which will be doing later on. What we are doing right now. So currently we recently received further funding to develop this as a model from the National Center for Replacement and Refinement and Reduction of Animals in Research, in collaboration with the Griffin Institute, which specializes in surgical training for pretty much all the surgeons. Every surgeon in their lifetime will have to go through this microsurgery training. And currently, microsurgery training is predominantly works on live animal, where you will be doing anastomosis, cut the vessels, and while the blood is flowing, you will be trying to suture them and all. That requires a huge amount of animals utilization in this case. So we collaborated with Griffin to look at whether we can create a synthetic model for them. And the people who work with hydrogels, they will understand it's quite difficult to suture hydrogels. But what we have done or what we have achieved so far allows us to suture them, put simple stitches on them. This is a microsurgery setup where you are going to be using very thin needles and micro fibers to put them together. And this is also a prerequisite for us to go for an animal study later on, if we have to validate this into a live animal to see that this is actually working. So it falls quite well within our timeline of development. What we are doing, again, we are also looking for cardiovascular disease modeling, and we are actively seeking collaborations in this area, where the collaborators could be interested in understanding the multicellular interaction between smooth muscles and endothelial cells, where there are a lot of other issues such as wall thickening of the blood vessels, which leads to various leaky blood vessels. Along with that, if you have osteoporosis or aneurysm, these are other specific diseases. So we're quite open for collaboration in this area, where we think these tools can be quite useful tools to study some basics about the cardiovascular diseases, which we currently don't understand. Further, we are moving to create some vascularized tissues. So I will just quickly show you the picture instead of going through pretty much everything we got on the screen. Is So what we have here is a block of a tissue, and you can imagine that tissue is filled with different types of cells, and then you have a blood vessel running through it. So this is a simulation of that, which we bioprinted, but essentially it shows you possibilities of what we be able to achieve by taking all these technologies together, where we are going to create a microchannels, and we are going to perfuse them with different blood imaging liquid, and the rest of the block is actually filled with types of cells we might be interested into, creating a vascularized tissue construct. Apart from that, there are some very important parameters which we are looking at in terms of progressing this technology, which is improving the properties of the inks we are developing, and which is actually quite important for progressing and tailoring them for various different types of tissues. And you can see here, some of my students are actually been looking into handling them, quite roughly handling them, printing them, roughly handling them, because these are the conditions which some of these devices will be faced in a real life application intraoperatively. So we are simulating them like exactly what we did when we are suturing them. In addition to that, we're also looking into transportation and storage. We think that transportation and storage is the key for biological tissue sampling, whether we are looking into clinical translation or even collaborating with people for disease modeling. This has a direct impact on the pathway to the market, the interventional method we are going to use intraoperatively, and overall, this will come down to the cost. So this is what we did. We actually printed our material and then freeze dried. And later on, again rehydrated them back to see if they actually retain their shape and they preserve their shape. And this is a graph which shows this is printed and then this is rehydrated, showing that they actually retain their original shape and the size, which is quite important because now that opens up lots more opportunity in terms of low cost storage and transportation for us, allowing us to collaborate quite widely with different groups. With that, I'd like to acknowledge all the people who have actually contributed to this work. We have a long list of clinical and academic collaborators all over the world. And most importantly, the people who funded this research, the MCR3 and the Wellcome Trust. And thank you very much. I'm happy to take any questions.
Is there evidence that 3D bioprinted tissues are more accurate than organoids? And I think further down, he qualifies that question with meaning a more accurate as a model for drug screening. I think you're on mute, Deepak. There you go.
Thanks, Mike. Thanks, Malcolm, for asking this question. Mike, can you hear me clearly?
Yes, I can hear you fine.
So I think it's a good question, Malcolm. Technically, there is no comparison. No one has done the comparison. The way I look at the bioprinting technology, it itself is a assembling technology. It allows you automation. The organoids can be a building block for bioprinting too. So instead of printing and mixing cells together, you could actually have organoids assembled and then printed to build 3D geometries. Obviously, there is a scale difference. So when we are using organoids, we are actually at a lower scale. You're looking at cluster of cells, which you defined. And in bioprinting, we are assembling them to go beyond few micrometer. I hope that answers your question.
Okay, thank you. Don't have a name here, but the question is the wall thickness, you stated it's 15 micrometers, but it looked a bit thicker in the picture. Is it sometimes thicker than 15, or what is 15? No, is that the average, or what's that figure?
It's defined. It's 250 micron. So that defined by the needle we use. Okay, so I would possibly direct them to look at our publication where they will be able to see the original picture with a scale bar. That will clarify. You can define those thicknesses based on the coaxial needle we use. So if you want to go thinner, you will actually design your needle in a way where you get the thickness lower than that. We choose 250 is because our future work which we are planning is to look into animal surgery to see if these things are viable. Well, they actually perform as well in a preclinical animal studies before going for a human trial. And the wall thickness of the rat models, they are actually 250 micron. So that was our justification for going with 250. But obviously you can tailor them. That's one of the beauties of bioprinting or 3D printing is very much of custom designing.
Okay, that's great. Thank you. Isra Mare, hopefully I pronounced that correct, is asking, hello, thank you for the great talk. In the bilayer blood vessels, were the endothelial cells and smooth muscle cells combined to their layers? Was there any migration observed? Also, did endothelial cells form a single monolayer?
Yeah,
there's quite a few questions there.
That's actually a good question. So we were quite interested to know. So Isra, what we've seen is there is some migration in the initial stages where you can see a diffusion of cell, which is possibly unavoidable. These cells are talking to each other. But as you start maturing, I think they clearly knows what their boundaries are. After 7 and 14 days, these cells start expressing and depositing collagen 1 and elastin. And I think this is where then the distinction between two layers actually become very obvious. Again, you can see the original picture in our publication.
Okay, thank you. So another question, could you elaborate, this is from Anonymous, could you elaborate on how this can be specifically applied for disease modeling?
Yeah, so I have been talking to some of our basic science researcher and cardiovascular team within UCL, and they were quite excited because there are issues in terms of leaky blood vessels. One of the examples where you need to see that the thickness and the wall thickness of the blood vessel can change based on the disease type. Currently, we don't understand the pathology. We don't understand why they happen. If we have to simulate those conditions, the only option you got is animal studies. You don't have anything in between. Obviously, we know there is a difference between animal pathology and human pathology. So having them based on patient's own cell and creating this vessel for a personalized patient specific application is quite feasible with this technology now. And you will be able to flow through your simulated liquids or drugs to see how those drugs respond to these different cell interaction, whether you are able to fill those gap between the leaky blood vessels. Some of these issues are also being highlighted by my colleagues into cancer research, right? Along with the block of the tissue I showed where we actually had a blood vessel running through. So the reason we are doing this is because we want to look at developing a co-culture model where it's being self-sustained in terms of its viability. Because most of the stuff in general in regenerative medicine, one of the biggest challenges lack of vasculature. So the work we are doing is possibly still at a fundamental level. We still need to do a lot more work to refine this. There are a lot of questions we have still. We can't say that this is a final model, but this will be the stepping stone to get those vascularization within the tissues. And when you have two tissues next to each other and the blood vessels are running through, you get those distinctions, which sometimes you are not able to do by using any other in vitro system at the minute. That's where I think the advantages will come in. The other one is these vessels, they have their physical properties, they have their elasticity. If you use a plastic tube or a coating or otherwise you use organ on chip model, you don't get them. You are just having a layer of cells, but they don't have any physical functionality and physical dimension. In this case, you are getting that real life dimension to it. So I would say it's a slightly higher scale than what you have currently. So there will be a lot more things we need to develop using this technology.
Great, thank you, sir. Marcio asks, which kind of growth factors can you use in pods for smooth muscle cells and for endothelial cells? And how does the delivery system work?
Okay, so I think for the pods for smooth muscles, we use TGF beta, and for endothelial, we use VGF. Uh, what our experience with the pod system was is actually doing a slow release. I think the mechanism itself, Mike is the expert. I think he'll be able to answer that. The mechanistic wise, we really were interested to see if we can actually do in situ differentiation, because currently you have two choices. One, you expand the cell in vitro, and then you combine them into a tubular structure. How about I start with a stem cell and don't worry about differentiating or spending time in the laboratory? Because if I differentiate them into tubular structure by starting with the stem cell itself, I'm saving about 1 week of my time. And the maximum cost we occur is mainly culturing these cells where we are adding a lot of growth factors into our media. We change our growth factor media pretty much every 3 days, right? We don't know absolutely whether we fully exhaust those growth factors. What we notice by using these systems is that we are not feeding new growth factors to our systems and to the cells up to 14 days, which itself was quite interesting because now we can save a lot of time and as well as money, which is where the growth factor costs us a lot. So there were particularly advantages. This is what draws us to think about a delivery system. And this seems to be quite conducive for the specific application we used.
So maybe I can just add, in terms of the way it works, it's a responsive delivery system. So the crystals, they're submicron size, they're about a micron across, and they are 100% protein, and they're a mixture of a scaffold protein and the cargo protein. And when you put them in the presence of proteases, the scaffold protein starts to break down and it releases the cargo protein. And as Deepak says, that happens over a period of about a month. So it's a nature mimetic system. Be very happy to answer more questions if you want to contact us through the website. We can tell you more information about the technology. But I'll go on to the next question, which is from Rachel LeBlanc. And Rachel's asking, can you explain how the cells were incorporated into the bioink?
They were physically mixed. So we used them different ratios. We did have to do optimization in terms of what ratio of these pots will actually give you the effect you want. So there is a lot of optimization experiment we did. Details of this again in our supplementary data for our publication. I will put the details of our publication in the chat box so you'll be able to access it.
Next question, Igor is asking, what do you think about metamaterials for 3D bioprinting of blood vessels?
Excellent idea, I would say. Maybe if he's interested, we can definitely talk and discuss what type of metamaterials might be interesting for this application.
For my benefit, perhaps, Deepak, could you explain what metamaterials are?
I think I'm not the expert in metamaterials, so I'm hoping I will speak with Igor and get more ideas. But definitely, I think it will be something interesting to learn. See what these technologies can bring into the stuff we are doing.
Okay. Next question from Anonymous. Have you tested microfluidics with these bioprinted models, or were these models you showed immersed in a bath of media? Was the media common to both cell types?
Yeah, so first one is, no, we didn't actually tested this microfluidics. That's our next step. We will actually use a large-scale bioreactor, which we have in the lab. Were the model were immersed? Yes, they were immersed in a media. And this is done in a static condition. So all the culture conditions were static. And that's the reason we are quite interested to know how the dynamic condition. This is how the original blood vessel is actually in a dynamic condition. So that's where now we are going for. Yes, the media was common for them. So the only thing which was changing is the pod system. And even though they're in co-culture system, the cell actually driving and utilizing what they need. Which is what we found is quite interesting. We don't have to fiddle with lots of parameters.
Thank you. Mohammed is asking, he says, hello, Professor. Thank you for a nice talk. If we use a hydrophobic component in printing, will rehydrated scaffolds attain their shape similar to post-printed scaffolds after freeze drying?
Maybe not. Maybe not, because you're changing the material. It's not the same way of absorption. And the reason they're changing and getting their shape back is because they are hydrophilic and they're able to reabsorb the water or the liquid. If you got hydrophobic, it will not absorb. So it will be a balance between it. So you might have to optimize those conditions if I had to be absolutely technically correct.
So best to try and avoid hydrophobic components if you're going to freeze dry.
I don't think freeze drying will going to have an impact. It will just stay as it is. It's just a rehydrating which will have an impact.
Good. Next question from an anonymous attendee. Nice work. 2 questions. How long will it take to get enough adipose tissue stem cells to complete one full vessel?
14 days.
I'll let you answer that first.
14 days. Right now we are taking 14 days. So 7 days, within a day, we get assembly. 7 days, we are seeing all the maintenance of. So technically the minimum required blood vessel is ready in 7 days. 14 days is where we give them a bit of more time to mature it, where we can start seeing elastin and collagen production. So right now I'll be very confident to say 14 days. But with dynamic system, we hope that we might be able to accelerate that process.
Okay. I can see, Mohammed, your hand raised. If you could put your question into the Q&A and I'll read it out if you could do that. The next question is, have you considered, this is again from the same person, have you considered the effect of shear stress of blood flow on the endothelial cell activation? Are you using pumps for flow?
Yeah, I think I clarified that earlier, is we are not using a dynamic condition so far, but this will be the next step for us. It's an important question.
Okay. Another question from another attendee. How efficient is the use of mixer nozzles in bioprinting?
We have not used mixer nozzles, so it will be difficult for me to answer that question.
Okay. So what do you think is the timeline for actually translating this kind of technology from the lab and actually getting it into the clinic? How far away do you think we are?
I think we still need to do quite a lot of work. We still need to do optimization like dynamic conditions. Because I think realistically looking into translation, when there is a medical need, the patient need these devices quite soon. We are talking in days, not months. Right now, the technology, what we see or people are actually publishing, it takes up to from starting getting cells to fully manufacturing can take you up to 2 months. So that's not a viable solution. What we managed to sort of find out whether there are alternative paths to shorten that process. Now, using adipose-derived stem cell as a source, we are able to sort of condense that down to 15 days from 2 months. So we think that's a step change. It's in the right direction. There were quite a lot of thinking about it from regulatory terms, from access terms, from patient terms. We know that these cells can be taken from the cells quite easily. We were quite focused and very consistent that we want it to be autologous source because of immunosuppressions and other issues related with transplantation. So now we are getting them up to 15 days. I think there will be work required for us to see if we can squeeze it down further. That's an ambitious work. And once that work is done, we have to do some animal studies, which will be required by regulatory standards. And once that's done, I think we'll be in a position to go for a human trial.
Okay, great. So a lot of work to go yet.
And still a lot of work, yes.
Yeah. So a few more questions have come in. Yesim Erdogan says, thanks for the excellent study and presentation. I wonder how you manage injecting cells into blood vessels without dispersing it into hydrogel.
So we simply inject them. We didn't find it. It's an important question. I know this is a technical challenge because most of the process, it seems quite easy when you inject things will just work. I think it's all about the bioink itself. The bioink itself, what we produce has taken care. We thought through that process to capture these cells on the inner wall in a monolayer. So we are simply injecting them and putting them. We do have a plan to simply print all the cells together. But then that becomes the printing technology limitations. That if you want to get a monolayer of endothelial cells, I don't think there is a nozzle small enough which will allow me to get the monolayer of the cells inside. So right now the coating seems to be working quite efficiently.
Great, thank you. Javier asks, thank you for the talk. Did you observe any contraction of the vacuum biomatrix mediated by the cells? If so, how did you manage to avoid it?
We don't see any contraction. Actually, we would love to see contraction. It's not a wrong thing for it to contract. This will obviously has to happen because the natural blood vessel will essentially contract and the cells will eventually contract it when they're fully mature inside a conduit. So we are actually hoping that we will see this, that phenomena. So far, we are not seeing any of those effects.
Thank you. Smruti, I hope I'm pronouncing your name reasonably well, has asked, how did you fabricate the Y vasculature structure which you demonstrated for simulating the dye?
How did we fabricate a bioprinting? So we bioprint.
Yeah.
Yeah. So
Bioprinting.
we use sacrificial gel inside a block, and we printed that Y structure inside.
Okay. And Rachel is asking, is the goal of the material to degrade and be replaced by regenerated tissue?
Correct. That's correct. Yes, we do not want that material. We want the cells to take over. And that's one of the difference between other technologies. And that's, again, possibly one of the limitation of bioprinting is you need a lot of cells. We are loading them with cells. so that the cells actually take over as soon as possible. And this material itself is just a scaffolding material and eventually is supposed to leave. So we are looking for it will actually disappear in 2 to 3 weeks time.
Fantastic. So there aren't any more questions at the moment, so we'll leave it there. Just